An individual has evolved a number of mechanisms, their immune system, to detect and destroy cancer cells. And the only way that a cancer cell is able to survive is if that tumor cell has evolved ways to get around those mechanisms. So one of the ways that it does that is it alters this packaging material through these chromatin remodeling complexes. And those are the discoveries that we observed and we published on over the last couple of years. We've then, through collaborations with researchers at the University of Minnesota, have identified small molecules that target some of those chromatin remodeling complexes. And we've been able to show in unpublished preclinical tumor models that those small molecules do work. What we want to do is we want to develop these drugs to a point where we can actually use them in people. Because funding is so low, the government agencies and the reviewers there are reluctant to fund early stage investigators, which are a little bit more risky for the government. So basically, only the top 8% of the grants will get funded. Bridge funding is really essential to keep the cycle of funding and publication going. If you're in a stage in your career where a grant ends and you need some time to get the final publications out, without bridge funding to do that, those publications won't get out into the literature, and then you'll have a gap in your publication record, which will make it almost impossible to get federal funding. The Virginia Moose Association provided that funding for our lab to give us this opportunity to continue the discovery process, to continue generating preliminary data, to continue to publish papers. All of that was essential for us to be competitive for these federal grants. With those research funds, we were able to generate a lot of preliminary data, and some of that preliminary data actually went into a grant application in which we scored a very favorable score recently, and it will likely be funded by the National Cancer Institute.